Good morning, everyone. It's my distinct pleasure to introduce our first keynote speaker. Dr. Barney Graham is the Deputy Director of the Vaccine Research Center at the National Institute of Allergy and Infectious Disease. He got his bachelor degree from Rice University and his medical degree from the University of Kansas. He was on the faculty at Vanderbilt, where I will point out to the trainees, he obtained his PhD degree while working as faculty member. He led the first human trial of AIDS vaccines there and moved to the NIH in 2000 to be one of the early members of the Vaccine Research Center. He has been a leader in viral immunology and vaccinology over his career, co-author of over 500 publications, where he's dealt with vaccines and monoclonal antibody development for a variety of diseases. And in line with what we're facing currently, he will tell us about vaccine development for COVID-19. Dr. Graham. Oh, and please, I encourage you to add your questions in the Ask Your Question box on the right side of your screen. Dr. Graham.
Well, thank you and good morning. It's my pleasure to be here and I appreciate the invitation. I'm going to talk to you, tell you the story about rapid coronavirus vaccine development, but also try to tell you some of our thoughts about how this informs pandemic preparedness in general and how it may influence the future of vaccinology. So just by way of disclosure, I'm an inventor on vaccine patents for a variety of viruses and also for monoclonal antibody therapy. I work at the Vaccine Research Center on the Bethesda campus at NIH, and this program, the VRC, was established about 20 years ago, specifically to develop an HIV vaccine. And even though we haven't been successful with that yet, many of the technologies that have been driven by this process have been applied to a number of other unmet needs, including emerging infections listed here. And we can do this because we're privileged not only to have this basic research building, but also about 100 engineers involved in process development, another 100 or so in pilot manufacturing, a self-contained clinic, and a laboratory devoted to good laboratory practice analysis of the clinical samples. It's run by this relatively small group of principal investigators and program heads directed by John Mascola, pictured here in the middle. And over the years, we've used a number of different approaches to deliver vaccines or immunity, including nucleic acids, which I'll discuss today, but also vectors, virus-like particles, proteins, including self-assembling nanoparticles and monoclonal antibodies, and have used and distributed these products around the world, including in 2018 when the monoclonal antibody for Ebola, NAB-114, was used to effectively treat Ebola and now has become a licensed product, Ebanga. So we're going to be faced with emerging infections going forward, and I think we've learned over this last couple of years that even though they're effective, the traditional approaches to outbreak management are somewhat limited. And fortunately, because of all these new technologies that have been developed, especially over the last 12 or so years, it has allowed us to take a more engineering approach to the problem of vaccine development, making it more modular and maybe putting it on a more dependable timeline. And so if you look at the history of vaccine development for viral diseases, at least, I've listed here the antiviral vaccines that we have available. Many of them occurred because of changing technology, opportunistically taking advantage of those technologies. And so the first two, smallpox and rabies, were really just done by the will of Jenner and Pasteur, and viruses grown before we knew what a virus was in animals. The next two, yellow fever and influenza, came because of the development of egg cultures. And the next cluster came because of the development of cell cultures, and ability to grow viruses to very high titer, either for live attenuated or inactivated approaches. And then the next several were a product of molecular biology, making either recombinant proteins, reassortants, molecular clones, learning how to grow cells in bioreactors now at high density, giving you the ability to either make a lot of virus or a lot of protein. And then recently we had the vector-based approach that I consider in more of the new generation or next generation of vaccines for Ebola, the vesicular stomatitis virus recombinant. And we thought that maybe RSV would be the next big thing in the new technologies, but using structure-based design to make a better respiratory syncytial virus vaccine. But coronavirus jumped up in front and not only used structural biology and gene-based delivery, but a number of other new technologies that illustrate how we are in a different era of vaccinology. So these technologies can roughly be divided into things that improve the precision of antigen design, like the structure-based design, being able to display on self-assembling engineered nanoparticles, being able to define antibody lineages. This all gives you better precision in either the antigen design or what you're targeting. And then the others improve your speed, speed for manufacturing, speed for analysis, animal model development, and so on. And these roughly correspond to the need for pandemic preparedness and pandemic response. You need time and a lot of time ahead of the crisis for preparation, for precision antigen design, and you need speed to apply those designs in the time of crisis. And we've thought over the last several years of ways of doing this in a more proactive way, and have proposed something we call the prototype pathogen approach for pandemic preparedness, and divide the 26 and now more viral families known to infect humans into their entry mechanisms, the class 1 fusion proteins, class 2 fusion proteins, and so on, and the non-envelope viruses. And if you could set up a system over some long period of time, combining intramural and extramural resources for research and development and solving structures and doing all the things you need to understand how to rapidly make a vaccine, and support those with core functions that could do the development part and product development along with the basic research that's needed, we think that you could come to a better place. And so the proposal is to look at those 26 viral families and some of the genuses of what would be about 120 viruses, develop about 30 prototype vaccines for about 30 prototype viruses through phase 1, and develop the others, at least through animal testing, to be better prepared for the future. And these are consistent with some of the priority lists that have been put together by WHO and CEPI, but it's a more systematic and comprehensive approach to this problem. Some of this thinking began when we had a breakthrough on the respiratory syncytial virus and were able to capture this pre-fusion structure of RSV-F glycoprotein in around 2013. And not only were we able to capture the structure, but it revealed this site of high neutralization sensitivity, new epitope at the apex of this molecule that doesn't exist on the post-fusion molecule, that spontaneously flips into this 6-helix bundle conformation, even on the virion surface. And you see the red surfaces are lost in this structure, so you don't have that target. And it explains why over the last several decades, 5 large vaccine trials focused on the F glycoprotein, all in the post-fusion conformation, could only boost neutralizing activity by about 2 to 3-fold. But if you can take this structure, learn how to stabilize it with trimerization domains at the C-terminus, internal disulfide or cavity-filling mutations, and then look at that now as an immunogen, you can boost neutralizing activity of 16-fold or more. And that turns it into a real vaccine candidate. And just to make sure you understand the dynamics of these entry proteins on viruses, I'm showing you a cartoon at the bottom where the virus is below, the host cell is above. And these fusion proteins, the class 1 fusion proteins that are on many of the envelope viruses of concern, begin in one conformation, grab the cell, pull the helical bundles back together, and end in this stable post-fusion conformation. You obviously want to target the functional part of the protein. So these class 1 fusion proteins, as I mentioned, this is the RSV on a pneumovirus. This is a para-influenza virus fusion protein structure, and they are characterized by these fusion machines, and it's on other envelope viruses like influenza, HIV, Ebola, Lassa, where you have a capping mechanism, in this case, covering up the fusion machine. The cap needs to be removed before the fusion protein can rearrange, and they're all organized in about the same way with this endoproteolytic cleavage site, a hydrophobic fusion peptide, and this is what then triggers the unraveling and pulling back together these heptad repeats onto one another. And coronavirus is another viral family with a class 1 fusion protein, and it's got both the cap and the intervening segment on top of its fusion machinery. So back in 2017, we had some insights onto how to stabilize coronavirus proteins and paramyxovirus proteins, and started a collaboration with Moderna, an mRNA company that can do rapid manufacturing, with the intent of demonstrating a proof of concept for this prototype pathogen approach for pandemic preparedness, and using Nipah virus as the paramyxovirus prototype and MERS coronavirus as the coronavirus prototype. Paramyxoviruses I'm showing you a cartoon here. They have both a fusion protein and a major attachment protein that could either be called G, HN, or H, depending on its hemagglutinin properties. And we can take the post-fusion or stabilize the pre-fusion form of the F, which we've shown to be more immunogenic, or independently take the stalk that's usually a tetramer, a dimer of dimers, as an immunogen. The problem with paramyxoviruses is you don't really know if the fusion protein or the attachment protein may end up being the better target. And so ideally, you could have both elements as part of your vaccine. So we have shown that the pre-fusion is more immunogenic and induces better neutralizing activity than the post-fusion form of the molecule. And combining the idea of the G multimer with the pre-fusion through this fold-on complex, we now have a chimeric molecule that combines the pre-fusion structure with individual monomers of G at the C-terminus. And for Nipah, we've shown comparing RNA to protein, comparing the pre-F alone, the G alone, or the pre-FG chimera, that inducing antibody to F is equally good in the chimera as it is to pre-F alone, and both the RNA and the protein. Inducing antibody to G is as good in the chimera as it is to G alone, and even neutralization is equivalent, but now you are targeting more surfaces and more epitopes. Maybe the biggest advantage of the chimera is that in T cell responses, using the AIM assay, looking at CD4 T cells, you get a broader, a bigger response to the chimera, especially in the T follicular helper cell compartment compared to G alone. At least in this hybrid mouse model, the T cell responses, both the T follicular CD4 cell targets, are more represented in the F molecule than they are in G. And the same thing is true for CD8 T cells. So you're improving the overall antigenic content for both antibody response and T cell response by using the chimeric molecule. And we've now shown that the mRNA can protect ferrets from a lethal challenge, and a clinical trial with this mRNA is going to start at the beginning of next year. The work on coronaviruses was started back in 2013 and 14, and eventually we were able to show the prefusion structure of spike on the endemic coronavirus HKU1, shown here. And when we had that structure, which was inherently more stable than the other spike structures for MERS or SARS at the time, we were able to find, in collaboration with Jason McClellan, this 2-proline substitution at the top of the central helix in that fusion machinery that prevents these helical domains from stacking and prevents the unraveling of the fusion protein, holding it in its original shape. Not only did it preserve the neutralization sensitive epitopes, but it also improved overall expression levels. The MERS, for instance, was expressed at 50 times higher levels than the wild type, making it a better construct for gene expression vaccine vectors. And we had tested this idea both by protein and mRNA with Moderna prior to the 2019. And so this is where we were at the beginning of the pandemic. And as you know, we've all lived through it. At the end of 2019, this virus was recognized as causing an outbreak in Wuhan, China. It's now spread around the world. COVID is caused by SARS-CoV-2 that infects the lower airways and the upper airways. But I'm showing you here an image of a ciliated airway cell in the bronchiole. And I'm showing you viruses coming out of this ciliated epithelial cell, some of them trapped in mucus. And if you enlarge that, you can see the abundance of viruses coming out of these ciliated cells. And if you enlarge it further, you can see that the virus is about an 80 nanometer sphere with anywhere from 25 to 35 spike knobs on the surface. And so this is the main surface feature of the virus, and it's the main target antigen for vaccine development. And as a testimony to all the new technologies that have been evolving over these last 10 or 15 years, there have been over 290 different vaccine development programs around the world. And now over 100 have been in clinical evaluation. This is going to change vaccinology from now on, because some of these different approaches to delivering the spike protein had never been tested in the clinic until this last year. So now there's going to be clinical experience and manufacturing experience with a lot of new vaccine technologies that had never been able to get out of the lab. And so it will change what happens going into the future. Over 20 of these have already been in phase 3, and there's at least 7 registered products. The nucleic acid, whole inactivated recombinant vectors have all, and recombinant proteins have all been authorized in different places. So it was really because of this prior work on coronavirus, informed by our work on RSV, what allowed us to rapidly make a design based on the sequences released in January of 2020, and then proposed the design to Moderna without any additional experimentation to initiate GMP manufacturing, also to then make protein for ELISA and structures, solutions, test immunogenicity, and within about 65 days, we were able to start the first clinical trial, and within 6 months, the phase 3 trial. One of the byproducts of this new technology is that the same thing you make for the reagents to do the vaccine program also allow you to use these reagents for antibody discovery. And starting with a collaboration we've had with AbCellera, giving them new probes for the new virus and some convalescent PBMCs, they use their sorting technology and microfluidic chambers and some of their sequencing technology, and we rapidly discovered a very highly potent neutralizing antibody we call NAB555, and that ended up becoming Lilly's bamlanivimab, which went into phase 3 within 5 months. So the same technologies that allow you to make vaccines rapidly also allow you to make therapeutic monoclonal antibodies rapidly. So once you know the structure, that the structure is right, by the end of January of 2020, Jason McClellan, again collaborating with us on this, was able to show the cryo-EM structure of the new spike. We could see that the mutations we used had it in the right conformation. It allowed us to confidently go on to help make diagnostic assays, as we did with CDC, the antibody with Lilly, and the vaccine with Moderna. And the work on the protein ahead of time also allowed the other red asterisk manufacturers to also use this protein or amino acid sequence in their vaccines, either nucleic acid, adenovirus vectors, or recombinant proteins. So people have been worried that this vaccine development process was too rapid and that it all happened in a year and it must not be safe if it happened that quickly. But I can tell you, or you've just seen how it didn't just happen in a year. It was based on 3 years of directed work on testing approaches using RNA for vaccines against paramyxovirus, coronavirus, and a public-private partnership. 8 years of work on defining atomic level structures of coronavirus spike and learning how to stabilize these spikes with generalizable mutations. 15 years of advances in protein engineering and platform manufacturing, other concepts on pandemic preparedness, and breakthrough discoveries on the structure of RSV proteins. These were all motivated by work and inadequate responses to pandemic threats from avian flu and chikungunya and MERS and so on, and 40 years of work on coronavirus biology and pathogenesis work on RSV, for instance, in that vaccine-enhanced illness. And everything was really based on the technology that was driven by HIV, efforts to make HIV vaccines. So the RNA, in this case, is a modified nucleotide RNA. It's the same kind of modified nucleotide that's used by both BioNTech and Moderna. It's 1-methyluridine in place of the normal uridine, and it's encased in a lipid nanoparticle comprised of 4 different lipids: a cationic lipid, pegylated lipid, cholesterol, and so this not only preserves the RNA, but helps deliver it into the cytoplasm of the cell, where it's immediately translated into a protein. And then that RNA is gone within hours. The lipids are gone within hours, and then the protein is expressed for several days. it is expressed for a relatively longer time because the 1-methyluridine helps avoid signaling through these TLR3 and 7 mechanisms or the double-stranded RNA detection systems in the cytoplasm. So overall, this would be considered a relatively low inflammatory event and allowing the cell to produce protein for a longer period of time. So having the RNA in hand and having the GMP process, we rapidly showed immunogenicity in mice first before we started the clinical trial, and then immunogenicity in monkeys. And so we have immunogenicity in the animal systems and humans, all well above what would be seen in convalescent individuals. There's protection of both lung and nose in mice and monkeys, and safety based on the paradigm thinking that you want a Th1 and avoid Th2 responses, as were shown to compromise cause the vaccine-enhanced illness syndrome and RSV. So we were able to show this is a very Th1 biased response when delivered by RNA, and also show that the responses, even in elderly groups of the older group and the people over 71, had responses comparable to the younger adults shown here, and similar types of T-cell profiles. So we started vaccinating frontline workers and others after we got the results of the phase 3 trial, which I'm sure everybody now knows. These mRNA vaccines were about 94 to 95% effective against symptomatic coronavirus disease. And so our co-workers and colleagues and students, and Dr. Corbett here, Dr. Kismikia Corbett is a fellow in my lab who is very involved in this process, Dr. Moscola, et cetera. And we've been celebrating vaccines with VIPs who came to learn more about it, our family members and church members. And the problem is, even though we're celebrating the vaccines, it's been hard to fully celebrate because this pandemic is continuing and it's been devastating, and it's caused a greater drop in life expectancy than anything since World War II or the 1918 influenza pandemic. That drop in life expectancy is even greater in racial minorities. We've had a loss of over 3 years life expectancy in Blacks, and Hispanics, almost 4 years life expectancy, and a total in the US overall of around 2 years drop in life expectancy. So even though the vaccines on the blue line is the first dose, the dark blue line is the second dose, even though the uptake of vaccines seem to have reduced the overall pandemic spread, as we immunized especially healthcare workers and elderly early on, this second wave right here was in younger adults. And now that they're immunized, it continued to go down. And so, but now, as you all know, there's variants emerging that are less sensitive to the overall vaccine immunity. And so we're having another fourth wave in the United States. And just to give you a sense of how these vaccines perform against some of the variants, there's some real world effectiveness data. This is not placebo controlled, but it's real world effectiveness data for the mRNA, AstraZeneca, and the J&J vaccine. And in the setting against the ancestral strain, you're still getting upwards of 90% real world effectiveness, even against symptomatic disease. But against these, the alpha, the first one that emerged out of the UK, the beta, the one that came from South Africa, the gamma, the Brazilian isolate, and delta that came from India, these different viruses have emerged, and although against these initial ones, the efficacy was pretty good, even against symptomatic disease, the efficacy remained high against severe disease. And so even though we're now having more symptomatic disease and what people are calling breakthrough, which are really just what we expect from a respiratory vaccine of infection in the upper respiratory tract, we're still getting relatively good protection in the lower airway. But we don't know how long that will last with waning immunity or with evolving, continued evolution of the viruses. So everyone's thinking about boosting, and we've just finished and posted a new paper on the bioRxiv, and I'll just briefly go through some of that data because it's similar to some data that Moderna has posted and DMID posted from the boosted clinical trial. So in this case, we had monkeys who'd been immunized for about 8 months. We boosted them either with the original vaccine or looked at a new beta strain boost compared to an immediate challenge. So what you see after the boost, either by giving the original ancestral strain vaccine or by giving a variant strain boost, is that against all these different variants, including Delta, which is in blue, you get a tremendous boost in neutralizing activity, even above what you had at the beginning after the initial primary series. And what you might see is that you even have the numbers have become closer. That same thing was true in the Moderna study, in which humans were boosted with either the original ancestral strain or the variant strain, the beta strain, and you boosted the original newt activity by 20-fold, 23-fold, but you boosted the response against beta by 32, and the response, the response against the P1 out of Brazil by at least 44-fold. And so you're boosting the variants even better than you're boosting the ancestral responses. And the same thing is true when you boost with the variant strain of 12-fold up on the ancestral strain, 35 and 27-fold up on some of the variants. And in the monkeys, when you do the challenge now with this variant challenge, it is quite resistant to the original vaccine sera. You're still getting very good protection in the lower airway, looking at day 2 or day 4 or day 7 compared to the control group. Less effectiveness in the upper airway, which is expected because the antibody doesn't penetrate the upper airway as well as it does the lower airway, but you're still seeing rapid clearance in the monkeys that were boosted. And this is by the PCR approach. The lower is by live virus titers. So boosting with RNA after about 6 months increases newt activity against all the variants, including Delta. Newt activity post-boost appears to be higher than it was after the primary response. And boosting, after boosting, there's less difference in the newt activity against the variants. So the conclusion of many of these animal and human studies we've done is that clear that there's a higher threshold of antibody needed for reducing viral load in the upper airway compared to the lower airway. That's why we're still having good protection against severe disease. It's relatively easier to prevent severe disease in the lower airway than transmission in mild disease in the upper airway. We think boosting will sustain a protection against mild and severe disease and also limit transmission. And we also think that it's likely that these are things that have not easily been measured and are not known for sure, but we think there's quite a bit of protection just from anamnestic responses that occur after infection that are fast enough to protect you from severe disease, and maybe even from some T cell mechanisms, because there's a lot of immunity even in people with low or negative neutralization in serum. And so getting back to this pandemic preparedness approach in general, in addition to setting up the long-term work we need to do on solving structures and understanding immunity in all these different virus families, there's some big cross-cutting biology and engineering problems that still need to be solved. I think the biggest of which is the problem of antigenic variation that still limits our ability to solve HIV and even make a better flu vaccine. Viral strategies for persistence, the problems of immunodominance and immune avoidance that viruses have that compromise our ability to change the direction of an immune response after it's been started. The ability to maintain selected B and T cell memory phenotypes, which is a problem in influenza, especially against some of the conserved epitopes. The ability to induce and maintain immunity in the right place, for instance, in the mucosa. And understanding the structure of particles, like the subviral particles of flaviviruses or the non-envelope viruses, the quaternary structures that make these particles is going to be important. And then understanding how to engineer these proteins and other approaches to get the cost down so that they can be used more broadly. And so, in summary, the proof of concept, we feel like we have a proof of concept for prototype pathogen approach for pandemic preparedness, and it's based on a lot of prior fundamental, basic, and translational research. Considering both precision and speed, one without the other is not that helpful, and also the critical need for pre-established public-private partnerships. We also, looking into the future, need to move some of these technologies from high-income countries to low and middle-income countries, and I think it's obvious to everyone that we need better vaccine and biology education among the general public so that there won't be so much resistance to these kinds of advances going forward. And just want to thank again the PIs that I work with every day. Jason McClellan, who's worked with us on all of our structural work, my lab group, especially Dr. Corbett, who was so critical for the coronavirus work over this last year and a half. Our academic collaborators at Vanderbilt and Duke, Mark Dennison and Ralph Barrick and Moderna, and a lot of other companies and organizations, DMID, Extramural Group at NIAID, was all very helpful. So I will stop there and take any questions.
All right. Thank you so much, Barney. That was really fantastic. You know, definitely myself have questions, and Bongsep has been texting me questions in the background here. One of the things that I always appreciate about keynote talks like yours at conferences are the perspectives that you bring to things. And one of the slides that I loved was an early slide where you tracked technology as an enabling driver in vaccine development from all the way from Jenner all the way up into the mRNA delivery technologies that we have now. And so one of the perspective questions that I have for you is, and I'm interested in your thoughts on this, is this the new era, right? Is this basically, can we expect that most newly developed vaccines are going to rely on this sort of mRNA technology and delivery? Do you think that's sort of where we are? Is that the future?
I think it is the future. Even for some of our established vaccines that we have, like the measles vaccine, that's a live attenuated vaccine, and it's extremely effective. but the technology that's being used to make that vaccine is over 40 years old, and the equipment's over 40 years old, and everything about it is becoming more and more difficult to sustain over time. And so even as we replace some of the existing good vaccines, I think it's likely that we'll be relying on some of these new technologies and new ways of making things. I hope it allows us to learn how to make things more inexpensively.
So there's some things coming in now in the chat. And so I want to, one of them is just a comment from April Clarkson, who's one of our high risk, high reward panelists coming up, and just says, thank you for all your efforts to help address the outstanding human health challenge and for the informative presentation. And then in terms of a question, one's just come in from Bader Jirai at University of Delaware. It says, thanks for the great talk. What is your opinion on the need for intranasal inhalable vaccine formulations compared to intramuscular, especially in the context of building mucosal immunity and IgA? Also, are the current vaccines showing robust IgA responses, and how do you think that will affect responses versus variants?
Yes, thank you for that. And you know, one of the reasons that a lot of the mRNA is being used right now is because it is the fastest thing to do, because it's synthetic chemistry. It doesn't require bioreactors or cell culture or anything. Even the platform technologies of the vectors requires expansion in cell culture and bioreactors. So mRNA is being used in part because it was fast, and we only know how to deliver that now parenterally. And many of the other things that are fast are given parenterally. It is not fast to learn how to grow virus or make it attenuated as something you could use as a nasal delivery approach. And so one of the big things I think that's needed in the future is, how do you deliver, for instance, self-assembling nanoparticles for influenza or other respiratory diseases intranasally, and make them and help them get through the mucociliary blanket and get into the right compartment where they can make the right mucosal immune response. Because if we have to go fast, using the live virus vaccine is not the fast way to do it. And there are new emulsions that may be useful for delivering things, proteins mucosally, or we may be able to aerosolize mRNA, but it really hasn't been advanced well enough yet. And there's other kind of risk factors for just putting vectors and aerosolizing those. So I think there is a lot of work that needs to be done on mucosal immunization, especially for respiratory disease, which would give you a better way of controlling upper airway immunity and transmission. These vaccines do induce IgA, but it's monomeric IgA, it's serum IgA, and it doesn't translate into what you would like to have as secretory dimeric IgA coming out of mucosal secretions. And whatever gets into the airway is transudated. It's not secreted in the way that you're thinking about. So we do make IgA, but it's not a particularly useful type of IgA.
That's really interesting. So April Clarkson has another question sort of related to this, but do you think this technology will now be deployed for addressing RSV and other respiratory viruses?
Well, Moderna just got fast track designation from the FDA for its RSV vaccine concept. They are delivering the same F protein that I talked to you about earlier, using a second generation set of mutations to stabilize it. And the initial data that's now been made public looks really promising. So it looks like we'll at least be able to deliver the RSV-F by mRNA if that's needed. And I think that would be a good approach, especially for young infants. I don't know if it'll boost as well as protein for maternal immunization in the elderly, which is being advanced now by other companies, but it looks like it may work for RSV. There's other approaches. There's other pathogens that mRNA is being used for. As I mentioned, we're using it for Nipah, and that looks promising. Moderna has a CMV vaccine that looks promising. I'm very hopeful that some of the influenza antigens that we're making can be delivered by mRNA. And so it is a technology that's going to solve a lot of things, but it's not necessarily going to solve everything. And I think the mucosal delivery may take a different type of technology. The boosting of the pre-existing responses may do better with other technologies, but for speed and for some other diseases and for priming, mRNA is going to solve a lot of problems.
All right, so I'll ask one other question, unless others arrive in the chat, and this is Bongsup's question. And Bongsup texted me this and said, why don't we ask him, what can you tell us about the Lambda variant and vaccines and just variants in general, and sort of where we're going with vaccine efficacy related to these new variants of COVID-19 virus?
Right. Well, this is an RNA virus, and we expected it to evolve as it is. Coronaviruses are really big viruses. They have a 30,000 nucleotide genome, and they have an editing function in their polymerase that most coronaviruses, that no other RNA virus, at least that I know of, is known to have. That was discovered by Mark Dennison at Vanderbilt. And so coronaviruses have an editing function that is used to hold on to mutations that help the virus. And so I think most of the mutations and variants that has happened so far have been to improve the transmission and infectiousness and ability to grow in human tissue. And that's why the Delta variant in particular is growing to higher titer than the other prior variants, and why the R naught, or how many people does each infected person infect, is going from about 2 up to about someplace between 5 and 9. And so the Delta variant is really learned how to transmit and grow and to maybe even go faster from upper to lower airway, which is probably why some of our younger people are now getting sick with this variant. I'm not sure how much farther it's going to be able to evolve to escape immune responses, and it'll eventually have to create a balance between transmissibility and immune avoidance. And I'm hoping that it can get trapped in there by the prior vaccine immunity, so that we get to a point where the number of strains is diminishing, or the number of major variants is stopping. But I think we really just have to wait and see what happens. Over time, as our immunity wanes and as the variants evolve, this is what they talk about on the news every night now, is when do we need a boost? Who's going to get the boost? And so as immunity wanes and the variants evolve, we probably will need boosting, but I'm trying to be optimistic that we may not need boosting very often, and that we're going to still have a lot of anamnestic responses that can be protective, and that we may be coming to a time when the variants don't come up quite so often. That's my hope anyway.
One last question, and then I'm going to kick this back to you, Alan. One of the perspectives that you shared that I really also appreciated as someone who teaches this was that the speed of the vaccine development for SARS coronavirus 2 really was built on 40 years of prior development. And so it's sort of an unprecedented speed from one perspective, but that was made possible by by so much other foundational work. But given that the platform is now, looks like it's safe and effective, do you think that in addition to being able to generate new vaccines more rapidly, that that approvals for vaccines will accelerate also?
I think these platform technologies like the ad vectors and the mRNA, that will make the regulatory process a lot easier going forward, because there's now hundreds of millions of people immunized and there's a huge safety database, and there are obviously some problems that have crept up as you immunize hundreds of millions of people, but there's at least a really good sense now of what are the safety parameters. And so it will make regulatory decisions going forward a lot easier. So if we have to change to a different sequence in the mRNA for coronavirus, I think those kinds of things will be relatively easy for the FDA to bridge using immunogenicity rather than another 40,000 person clinical trial. So I do think it will help things. I don't know if you can go now in with a mRNA for influenza and show immunogenicity, or for some other virus and show immunogenicity and say this must be good enough, because the cautionary tale is that, if you don't deliver the right protein, you may not get much protection. So the precision has to go along with the speed. So mRNA is going to help a lot of different things, but it doesn't solve everything. That's why I still think we need a lot more basic research on these other viral families in order to really have all the information we would need to make a rapid response in the future.
All right, I have one last question. It arrived in the chat while you were making this answer, and it's sort of related to the approval process, and it asks briefly, why so much caution for the vaccines to be injected into a younger population? And that's from a user named Minjay.
I think that there's not a lot of reluctance to go into younger children. The FDA is struggling with the use of emergency use authorization, which has never really been used for vaccines before. It's been used for drugs on occasion. The only time it's ever been used in a vaccine setting is when they extended the shelf life of an anthrax vaccine just to make sure it could be available if something happened. And so using a vaccine for emergency use, authorizing that, is a very unprecedented step for a vaccine. And so the criteria for being an emergency, balanced out against any potential side effects, there's just a higher bar there. And as you go down in age, even though this virus is causing more disease and more severe disease than RSV or influenza and other things that we're more used to, I would like to see children vaccinated myself, but having it done under emergency use, I think, is a hard decision for the FDA. And most of the companies have now taken their vaccine evaluation at least down to 5 years of age, if not down to 2 to 3 years of age. So there's no reluctance to test it in young people to get the safety profile. And I think once the vaccines are fully approved, that it won't take too long to then move the approvals down into younger children.
Great. Thanks, Barney. Alan, I'm going to hand it back to you to wrap up. We've exhausted the questions in chat.
Well, given the time frame, I think we'll call an end to this session. I want to thank Dr. Graham for a wonderful presentation, very informative, and great questions. Thank you all.